Good day, my dear postgraduate students. Welcome to a novel class on gene therapy. And I believe that these are more costly than the regular intact genes. You people must be knowing more about it, but then I will not be going into it, but rather this. Topics like this have been included in our university exams. Let us see what we can unravel. So it is obviously an MD postgraduate university question. Short or long doesn't matter. Again, probably some kind of suturing is being attempted at the DNA level over here. Gene therapy is defined as the introduction of normal genes into cells containing defective genes so as to substitute a missing product. This is one. So you are introducing something that is perfect and intact into a defective cell containing defective genes or to correct deficient phenotype. Some might not be obviously visible, but then those can be corrected. This can be a definition for gene therapy. But then there are certain exceptions. All that is being introduced into the body cannot be considered as gene therapy. And the exception that the authors give will be the bone marrow transplantation. For that matter, any organ transplantation. So we will have this in mind. Gene therapy is different, transplantation is different. The history. Something splendid that people have been doing. Martin Clint, 1890, was the person to first attempt the modification of the human DNA. Successful DNA transfer was then brought about by the National Institute of Health. Though this person attempted first, he was not very successful. At least that is what literature says. That is why you people should record your findings and publish your research. Therapeutic gene transfer was done by French Anderson. And between these years, more than 2,900 clinical trials have been conducted. And these are certain other researches that have been mentioned. There are lots and lots of it. You people can go to the net or to your journals. And these are supposed to utilize the adeno-associated viruses, AAV, and lentiviruses. After all, what is the purpose of all this? To cure or to treat genetic disorders. And in the process, these are various trade names that have been given to the various products that have been used across many a disease. For the time being, we shall be remembering these two. Adeno-associated viruses and lentivirus. One is for in vivo, another is for ex vivo. What are these two? We shall be seeing. There is a broad classification of gene therapy. One, it is non-specific. So it is too broad a term. Therefore, people wanted to very concisely bring it into a fold of definition, mainly for legal purposes rather than scientific purposes. The legal implications, as you people can imagine, can be very useful or disastrous. Imagine me trying to become the most handsome person in the entire world, and I accordingly take some. Or one of you wants to become the most brilliant human on earth forever, and then you attempted gene therapy. Still others, they want to have offsprings which are successful in all walks of life and they start attempting at it. Or somebody else, as in the movie Indran, etc., try at creating a disaster itself. So, it is controversial, just as in the case of RNA vaccine. Though this is not a gene therapy, it also came into controversy initially. Fine. Now, what is this? These terms we people should be somewhat familiar with. Just hear them at least. Gene therapy is the addition of nucleic acids. The gene augmentation adds new protein coding gene to a cell. So there is a new kind of slot that is given to a cell. Gene replacement therapy is one form of gene being replaced by another. And it can also be considered as gene augmentation. Gene silencing, on the contrary, is used for many a disease in which there is a causative gene behind it. Sometimes they can be caused by multiple genes. And the Journal of Law of Biosciences, see, it is not a journal of medicine. It is a law of biosciences preferred a narrower classification. Because tomorrow we can imagine the thousands of cases that are going to plague each and every court on earth. Now, what are the types of gene therapy? I would like you people to kindly by heart this. Basically, there are two types of gene therapies. One is called the somatic cell gene therapy. Somatic cells are cells that will be multiplying, but they won't be going in for meiotic divisions. They undergo mitotic divisions. So, they can be transferred to any of the cells other than gametes. Gametes are where there is a half division of the cell. Somatic cells are modified genetically to prevent a genetic defect in the offspring. So you can use this so as to prevent a genetic defect in the offspring. It can form a basis for clinical research and treatment. And certain diseases such as immunodeficiency, thalassemia, sickle cell anemia, cystic fibrosis are all being attempted at being cured by means of this. So there is a harvesting of the mitotic cells. Mitosis is important. Always the cell should be in a state of multiplication. They should not be stable cells or permanent cells wherein this is possible. But at the same time, they should not be and cannot be passed on to the next generation. Otherwise, it will become a movie like Indran. So, somatic cell gene therapy. So, this is important. Non-dividing cells, at the same time, cells which are capable of mitotic division for curing many a disease such as thalassemia, cystic fibrosis, etc. Now, germ cell line therapy or germline cell therapy. These are modified by the introduction of functional genes and they are used to correct a genetic defect. The change is heritable and it can be passed on to the subsequent generations. This is very, very controversial. You can treat a patient, but not the next generation that follows. And that is why many countries, such as Switzerland, prohibit this on ethical grounds. US permitted it for therapeutics, however. This is better tolerated and it has got less immune reactions when compared to the somatic cell gene therapy. So these two things we people shall and will memorize. Again, there are two things. The heart is here, which means we will have to by heart. There are two types of these gene therapies. One is in vivo and the other is ex vivo. In vivo means within the body. Ex vivo is outside the body. First, we shall say what is in vivo gene therapy. A vector or a virus is introduced into the cell. This is a very common thing that we always see in neoplasia and the other topics. You find that the gene or the DNA is affected by means of a virus. The virus cannot multiply. It uses your own chromosomes for multiplication. Therefore, it enters the cell, becomes part of your own chromosome or the DNA, multiplies, and then it comes out. In this case, the genetic material is passed into the patient cells by means of this vector virus. The prosthetic gene therapy has been aimed at covering all the physiological functions which are not normally carried. One example in this case is vision, wherein there can be a restoration gene therapy in patients with end-stage retinal diseases. So this is in vivo therapy. There is a material that is being introduced. Then what is ex vivo? You find there are certain therapeutics which are being used, such as R.A.T.S. In this case, it can be the patient's own cells, autologous, or it can be from a donor, allogenic. They are modified outside the body and they express a particular protein, chimeric antigen receptor. And these are being introduced into the host later on. The CRISPR gene editing is one. We will just remember this particular word alone, CRISPR. It is important. It has been used for gene therapy. Instead of a pure replacement, it enables the correction of a genetic defect that is already existing. So there are two types of it, in vivo and ex vivo. In ex vivo, you find all the process happens outside and then it is again being introduced into the body of the host or the cell of the host. This is a viral vector. You find that there is a healthy gene which is capable of multiplying. And the viral vector, it makes use of this as a vehicle. And then it is introduced into the cell or the tissue that is to be corrected. And this is a gene therapy in a host. So this can happen. There is a viral vector. And we have mentioned two viruses. I would like you people to kindly recollect them. And even before I go to the next slide, this can be limited by the viral receptors that are present on the surface of the cell. So on the surface of the cell, you find that there will be receptors for the viruses. Sometimes there can be too much of intercellular spaces. And even on introduction, it cannot be passed on from one cell to another. These are some of the limitations which we shall be having in mind. Ex vivo gene therapy. This is a beautiful chart and this is a reference for it thanks to Wikipedia. So kindly follow the sequence of my talk. There are copies of the therapeutic gene. The gene are inserted into the viral DNA. So this is one. And then they are put in for culture. At the same time, you find target cells are removed from the patient and the cells are grown in culture. So the viral DNA as well as the host cells are grown in culture. And after which you find that there is an alteration in the genetic sequence of the patient. And these cells which are modified are reintroduced. And inside the body, we get genetically altered cells to produce the desired effect. Forget all these things. Anyway, you can read it. Wikipedia, and this is the reference. So again, I repeated, copies of a gene, and then they are inserted into the viral DNA. At the same time, cells from the patient are being removed. Both of them are grown in culture, and then there is a modification, after which it is again getting reintroduced. This is called ex vivo gene therapy. Variety of gene therapy products. There can be the plasmid DNA, viral vectors, bacterial vectors also, not only viruses. Human gene editing technology has been used to disrupt the harmful genes, and patient-derived cellular gene therapy. Cells are removed, genetically modified, and then they are returned to the patient. So maybe it is something like plasmapheresis and then a reintroduction. So this kind of things we people shall be remembering. And you find that they are all limited by an organization called the Food and Drug Administration. And dear students, do you know who gives the license for the blood banks in your colleges and hospital? It is not the Medical Council India or the National Medical Commission. Try to find out. Now, what are the uses of the gene therapy? To replace the missing or the defective genes. To deliver genes that hasten the destruction of cancer cells. And to add positive genes that revert cancer cells to normal. Deliver viral or bacterial genes as a form of vaccination. It can promote or impede the growth of a new tissue. Deliver the genes that stimulate healing of a damaged tissue. So these are the various uses. I would like you people to kindly by heart this particular list. Suicide gene therapy. Suicide means you must be knowing about the suicide bombers. They enter a crowd and then they switch on the button and then there is a lot of death around. Similarly, you find that there is a gene called the suicide gene. And when it is introduced into this particular cell, it gets incorporated in it and it produces a toxin which can be destructive to the cell itself. It was used to treat cancer. And this is the reference again thanks to Wikipedia. There is an apoptotic chain, the cancer cell is getting killed. What is the principle basically? You find that the viral capsid, that is a capsule, has to be stabilized. There has to be very low immunogenicity. Otherwise, there is a chance of a hypersensitivity like reaction. It has to have the ability to transduce both dividing and the non-dividing cells. It has to penetrate both and integrate at a specific site. It cannot be wavering here and there. Sometimes you find when the integration is at a wrong site, the results can be disastrous. That is one of the disadvantages of gene therapy. Achieve long-time expression in vivo treatment. So it has to be not short-term. There are certain cases where it is short-lived, but it has to be a long-term. And these are the two adenoviruses. So they are used for in vivo delivery. And the other can be the lentivirus used for delivery in vitro. Even for immune deficiency, such as AIDS, you find gene therapy is now being attempted. Look at this one. There is the working copy of a gene that is getting inserted into a virus. So this is a viral cell. It is getting inserted. And the virus has got a property of going inside the host cell. It goes and infects. But what is happening here? There is a modified gene that has been inserted. Once it is there, now what happens is it goes and produces the stem cells and these are again returned to the patients. You are introducing a kind of a viral infection, almost like a vaccination. You are allowing the patient to create the stem cells against the virus and now this is being used as a form of therapy in immune deficiency states. In AIDS, this has been attempted. The same thing I have mentioned in the box. Kindly go through this. There are the viral vectors, as I mentioned. These can be the various viral vectors. Sometimes there can be non-viral vectors, such as the liposome, then gold nanoparticles, exosome, ferritin, etc. So they can be used as gene therapy drugs. And they will be modifying the various forms of the RNA or the plasmid DNA, etc., as well as the CRISPR. And as a result of which, they can be used against treatment or there can be for Duchenne's muscular dystrophy, for Parkinsonism, Mediterranean anemia, that is a thalassemia over here, glaucoma, etc. And this is a reference for this beautiful flow diagram. So kindly go through this one. Definitely, a little picture is worth a million words. Again, something that I cannot imagine of even seeing, leave alone. Maybe you people will become great one day and apply for the Nobel Prize. So in this case, you find that there is a viral vector over here that is being inserted. So it is an invive, and it can be injected in the region of the macula, retina, the choroid, the optic nerve. As a result of which, you find that there is some change over here. There is an expression of the RPE65 gene is maintained episomically, and the RPE65 protein is being expressed. So it can lead to some kind of a regeneration in a case of a macular degeneration used for treatment. Again, it is mentioned for sickle cell anemia. We are collecting the stem cells, and the stem cells are corrected. The beta globin chain is being corrected, and along with the viral vectors, it is being injected again, getting transplanted to the patient. Probably there is a kind of correction therapy. Abnormal hemoglobins you are removing, normal hemoglobins you are replacing back. A wonderful idea, at least for patients who are counting their days. And this again is the same. It can be a viral gene, there is a new gene, and there is a vital gene. The vector is binding. This is what I said. There will be a receptor site. So whatever be the virus that is being used, the cell should be having a receptor site. Only after which you find that can be an internalization, it goes in, and then there will be the various changes. RNA gets converted to the DNA, etc., and it gets incorporated into the host. In carcinogenesis also, this will be the story. After which there can be a multiplication, and only thing, it will not be an active infective virus, but an inactivated one. So the reference for this is over here. I would like you people to kindly go to. So this again is a little difficult. It is regarding the CRISPR and the Cas9. So you find there is a complex that is being formed, and it is being made to integrate within the cell. There are a lot of changes which are happening over here, such as the severing of a DNA, and ultimately you find different combinations occur. I do not know much about it. Rather, you people may be knowing much, but this is the explanation or the expansion for the various terms. For example, DSB, it means double-stranded breaks. So both the strands of the DNA are getting broken. And HTR is supposed to be homology directed repair. That is happening over here. NHEJ is non-homologous end joining. So there is something over here and something is getting joined. So these are various ones. HNH and ROOF-C. So these are over here, which are mainly belonging to the endonucleases. So this is literally beyond us, but we shall have a concept about what is happening, and definitely we will at least mention CRISPR and Cas9. And these are the various classifications regarding the gene interference, gene insertion. And a lot of proper names have been there to protect or to indicate the place where it has been developed. And what are all the indications? In this one, I would like you people to kindly collect what are all the uses of the gene therapy. Macular degeneration, hypercholesterolemia, Duchenne's muscular, spinal muscular, hepatic veno-occlusive, and lipoprotein lipase deficiency, thalassemia. At least this much I would like you people to remember, not the countries and the years. So the general applications can be gene editing, genetic diseases, viral infections, cancer. This will be more important. And what are the adverse effects or the shortcomings? One, they can be short-lived. So in that case, therapeutic DNA, it has to be functional. Cells must be stable. Immune response. Any foreign substance, it can lead to an immune response. Therefore, we must be aware of the effectiveness and the contradictions. Problems with vectors. Toxicity and inflammatory response. After all, whatever you are going to inject is going to elicit an inflammatory response, which can be again dangerous. Some disorders such as Alzheimer's, diabetes have got multi-gene disorders. So in this one, if you try a gene therapy, it might not be that easily successful. It can be incomplete or futile. Sometimes insertional mutagenesis in a sensitive spot, like a tumor suppressor gene, can lead to a change It can suppress this tumor suppressor gene, therefore it can lead to a tumor. Corrective gene therapy in X-linked severe combined immunodeficiency can end in leukemia. This is a story that is well established. Most people overestimate what they can do in 1 year and underestimate what they can do in 10 years. So probably in the long run, you people can do a great thing. Just like Bill Gates, I'm nowhere near his shadow, but then it is a kind of belated wisdom that I am rendering you my classes. Thank you for your patient listening.